Peter Langfelder. He is, of course, the brain and the muscle behind the WGCNA R package that we will teach during the course. So he maintains the R package. He has a background in physics and string theory, but he has worked now on genomics data for over 10 years. And he will start out with an introductory lecture on hierarchical clustering and branch cutting. And so he will talk about the dynamic branch cutting method, which he developed. And I think it's really the state of the art when it comes to finding modules based on a hierarchical cluster tree. And with that, I'll stop. Peter, take over.
All right, thank you. Can everyone hear me? Are the microphones on? Okay, so it's very nice to see a big audience that's still interested in our methods. I'll give a very brief overview of clustering and the dynamic branch cutting because it's not the main focus of these lectures. The main focus is really the applications. So here's the outline of my talk. I'll start with just telling you what clustering is, so you're not lost right from the beginning. Then I'll spend a few minutes on hierarchical clustering, which is a particular type of clustering, and then I'll tell you how to find clusters, what we call modules, in hierarchical clustering trees. And there are really two main ways to do it. The first one is a simple fixed height tree cut, which works well in some cases, but not in others. And the reason why we developed the dynamic tree cut was precisely that the fixed height tree cut doesn't really work very well in more complicated situations. So I start here. What is clustering? Clustering is a method that takes as input a pairwise matrix of dissimilarities between objects. So here I have, let's see if I can find the pointer here. There we go. So I have 10 objects. I call them gene 1 through gene 10. And there's a 10 by 10 matrix of numbers. And the numbers range between 0 and 1. And if it's a 0, it means the genes are very similar. If the matrix is 1, it means they're very dissimilar. So you can think of it as a distance matrix. And the aim of clustering is to find groups, which are called clusters, of objects that are closely related, of course, according to the matrix here. So the output of a clustering method is a vector, is a cluster label for each object. So each of these 10 objects gets a label, and the label could be numeric or it could be a color label. So here, these 4 objects have the same color, so they're part of the same cluster. And then these 4 objects here are also part of the same cluster. And I should say that the gray color here really means that it doesn't mean that the 2 genes or 2 objects are part of the same cluster. It means that they're unassigned. They're not part of any cluster. If you have a high throughput data set, especially from a biomedical research project, there are many applications that you can think of for clustering. So first of all, you could cluster samples, which are oftentimes patients, and this is usually used to discover subtypes of heterogeneous diseases, for example, cancer, neurological diseases, and so on. Now, instead of clustering the samples or the patients, you could also cluster the high throughput molecular phenotypes, so the measurements, which are usually either gene expression or methylation, proteomic, metabolomic, and I'm sure that there's other omics that I'm not even aware of. And the aim is to discover organization, so transcriptional or methylation or whatever the applicable word would be of the genome, of the protein complexes, and so on. But more importantly, the aim, especially for the correlation network analysis, is to identify the candidate key drivers. So if you have a disease and you want to find the genes or the proteins that you should focus on first in your further follow-up, you could use network analysis and clustering. Now, apart from these two main directions, there are other more complex and more complicated analyses that are possible, especially if there are multiple data sets available, which is these days pretty much always. Now, there are many clustering methods, and when I say many, I really mean many, as in hundreds or perhaps thousands. Some of the most popular or maybe just most classic ones go by the names of k-means, partitioning around medoids, known as PAM, self-organizing maps. There are model-based clustering approaches, and lots of other methods, and I really mean lots. So don't be surprised if you see that every analyst has his or her own favorite clustering method. That's just the way the field is. There is no clear winner, there's no clear loser, and there are lots and lots of candidates. What we usually use is known as hierarchical clustering, so it's one of the many clustering methods. And why we use it is because it's a good method for exploratory data analysis. And exploratory, we mean that you get a big data set and you just want to see what's in there. You don't necessarily need a ranked gene list with p-values and FDR. You just want to see how it's organized, what's in there. And why is it good for this kind of analysis is because it works well with high-dimensional data, and it provides a visualization of the cluster structure. And it does not require you to know anything about the data beforehand. In particular, it doesn't require you to specify the number of clusters, which a lot of these other methods do. So you have to specify that there are 10 clusters for k-means, for example, but not for hierarchical clustering. How does one use hierarchical clustering for cluster identification? It's really a two-step process. In the first step, one constructs what's known as a hierarchical clustering tree or dendrogram, and this is an example based on the 10 objects that I've shown you before. This is a hierarchical clustering tree. And then one identifies branches. So branches are really what you think of a branch as a tree. This would be one branch and this would be the other one. Okay, and we label branches by either numbers or colors. So this branch has color blue and number 2. So more precisely, the objects on the branch. And this branch here has color turquoise and number 1. Okay, so these genes are number 1. And the gray ones, they're unassigned, and we usually leave the color of unassigned object 0, and the label number is, the color is gray and the number is 0. So I'll show you a cartoon of how hierarchical clustering works. So we start, this is going to be on the 10 objects. This is simulated data, so I know what's going on. So we start with the dissimilarity matrix that's here represented as a heat map. So the red objects are the red entries are ones where the dissimilarity is low, the objects are close, and the white ones, like here, are such that the dissimilarity is high, so the objects are far apart. Okay? So in step 1, you find the 2 closest objects, and it just so happens that the darkest red color was here, and I colored it blue here, and you merge them. And so this is, I hope the people in the back can still see it, maybe not. So at the bottom of this empty field here, there's a little merge of the 2 objects, and the height, the y-axis level where the merge was drawn, corresponds to the dissimilarity that was in here. Okay? And the dissimilarity is grayed out. So we merge these 2, they're now one object. In the next step, we find the 2 closest objects again, which just happen to be these 2, and we merge them. And again, 2 closest objects, and now it just so happens that the 2 closest objects are this small cluster and another gene. Okay, so that small cluster gets merged with this gene here. And another, again, we find the closest 2 objects, we merge them, closest 2 objects and merge them. And so the tree grows until all objects are merged into the tree and the tree is complete. Okay, so this is the final clustering dendrogram. So what do we do next? Oh, sorry, I should say first that there are several versions of hierarchical clustering which differ in how they measure dissimilarity between an object and a cluster. So when you have two objects, you just have a number. When you have two clusters, or an object and a cluster, you have a bunch of numbers, and you have to distill that bunch of numbers into one single number. And methods differ by how they do it. I'm not going to go into the details. I'm just going to say that we usually use what's known as average linkage hierarchical clustering, where the dissimilarity of two clusters equals the average of the dissimilarities between their objects. So if you're looking at this cluster and this cluster, okay, you just take the average of the dissimilarities between the objects here and here, which would be the average of this part of the matrix. So it's pretty simple. It works well, but like I said, other people have other preferences, and so someone else may tell you, no, you shouldn't use average, you should use this other method, and so on. That's fine. If you're using R, which is what we use and what our tutorials will be in, hierarchical clustering is implemented in the function hclust, and there are several versions of the function. In the standard package stats, there's an hclust which works reasonably well, but if you have a really large data set or you want the function to really run fast, you should load the package fast cluster and use the hclust function from that package, which is what I use. The function has two important arguments. There is the distance structure that represents the dissimilarities between the objects, that's called D, and the method. And like I said, there are several methods, but we usually use average because it works well. And the result that the function gives you is the hierarchical clustering tree, or its programmatic representation, and you can display it using the plot function, or you can use it as input to other functions such as the tree cutting or branch pruning functions. Okay, so we have the tree. How do we now identify clusters in that tree? So if you look at this clustering dendrogram here, the tree, it seems, just by visual impression, that there are two big branches. Okay, so what are the methods that can identify those two branches? So if you have a simple tree like this, there is a simple solution. You pick a suitable constant height, which is this red line in the clustering tree. Here, the number is 0.97. And you literally take scissors, or you imagine you take scissors, and you cut the tree horizontally at this height, and whatever falls down is a cluster. So initially you get 4 clusters because there's 4 things that fall down, but then we usually impose a restriction on the minimum size that the cluster must be. The cluster must be at least a few genes big. We don't want clusters that are 2 genes or 3 genes. Actually, in practice, we want clusters that are at least 20 genes big, but the number can differ. Okay, so this method works okay, but it has very important drawbacks. If you choose the cut height differently, you will get different results. So if you move this line just a little bit up, closer to 1, you may suddenly find that these 2 branches form just one cluster. And that's not visually what we see. So the proper cut height setting is essential and requires an intelligent operator. It's hard to automate it. In general, each application will require a different cut height. And so it's hard to do a comparative analysis where you have several data sets and you want to compare modules and like, well, you know, if I choose the cut height this way, I get very good overlap, this way, very poor. It's not that good. Now, nevertheless, it's used, and we also use it when it's appropriate. So in R, you can use functions cut tree static and cut tree static colors, which are part of the WGCNA package. And the difference is that the cut tree static returns numbers, 1, 2, 3, and the unassigned label is 0. and the cut tree static color returns color labels, which correspond to the numbers. So number 1 is always turquoise, number 2 is always blue, and so on. You can use the command help with the function name to see the details and what arguments the functions take and so on. Now, the simple solution is nice, but it doesn't always work, especially if you have a complicated tree. So if you look at this dendrogram, there are several branches that at least look like they're distinct branches. So there's one here, there may be one here, and there's these, and then who knows, maybe this branch and this branch are separate. And so if you want to capture all of them, no single cut height will do. So what does it mean? Does it mean that hierarchical clustering is not good for this? Well, it does give you nice branches. So is there a method that could adjust the cut height so that it captures most of the prominent branches? And there was none when I worked on it, or actually the work was started by Bin Zhang, who was a postdoc with Steve before me. But he developed the first version, but I kind of didn't like his approach very much. So I thought, well, maybe there's a better way. So anyway, the two of us eventually came up with a dynamic tree cut. So if you, this is an example simulated dendrogram, but the important part is that the function kind of goes from the bottom to the top in a similar way as the dendrogram is constructed. And when it sees that two branches merge, which would be here, and it looks kind of at the branches and decides, using a variety of criteria, whether the two branches look sufficiently distinct. Okay, and if they look sufficiently distinct, they're assigned different color or different cluster labels. If they're not sufficiently distinct, then they're merged and the function continues upwards. Now, one thing that is fairly important and that people sometimes don't realize is that once you merge two big branches like these two, objects that are merged into the dendrogram above that could belong to either one of these two, and the clustering tree itself does not tell you which one it is. Okay, you might think, well, these ones are closer to this branch, so they should all go to here. But that's not true. Okay, it's just the way the dendrogram is plotted. You could switch these two branches on the plot. It would look just the same, and suddenly you would think that these objects here should go to the other branch. So the tree basically doesn't tell you where the object should go, and therefore we implemented an optional step that basically looks at each of these objects and looks at which one is it closer to using the dissimilarity information that's as input for the clustering. And so what can happen is that these objects, you can see here, this big branch is turquoise, this big branch here is brown, and the objects above that merge are kind of, one here, one there, one here, one there, which looks surprising, and a lot of people are uncomfortable with this. But the way I simulated the data, the function actually gets the simulated assignment pretty well. It's not perfect, but it is pretty good correspondence between the colors here and the colors here. So it is an optional step, but it works pretty well. I call it a PAM-like step, partitioning around medoids, because you look at sort of the average distance between the object and the clusters, it makes the colors under the clustering tree look a little bit mixed up, and a lot of people think that this is a bug, but that's actually a feature. Now, if you're running R, the function, the dynamic tree cut method is implemented in the function cut tree dynamic in the package dynamic tree cut. So if you want to run it, you just say library dynamic tree cut and help cut tree dynamic. It takes as input the clustering tree. You need the dissimilarity matrix as well. And there are multiple options that fine-tune the behavior, the criteria for whether two branches are called separate or not, and options for the PAM stage. Now, when I say there's a multitude of options, there is really a multitude of options, but there are two most important ones that I want you to know about. The first one is called deep split, and I didn't come up with the name, so please don't ask me why it's called deep split, but it's a number between 0 and 4, and in the current version, you can also supply fractional numbers if you want to fine-tune it. It controls how finally the clusters will be split. And the second argument is PAM stage. This is just a logical false or true, and it turns the PAM stage on or off. So I'll show you what the effect of deep split is. This is real data. It's about 3,000 genes, if I remember correctly, from a mouse liver application. And so if you start with no PAM, so the more outlying objects are not assigned, and deep split 0, the function produces just a few relatively big clusters. Okay, although if a branch, like these small branches here, if it's really distinct, it becomes its own cluster even at deep split 0. If you increase deep split to 2 and then 4, the big clusters start splitting up into smaller ones, smaller and smaller. So I usually use deep split number 2 if you ask me for guidance. I think it's a good compromise between not losing small clusters and not splitting the big clusters too finally, where the fine splits may just be kind of flukes in the data. So this is what deep split does. Higher deep splits give you finer clusters. Now, what does the PAM stage do again? So remember that without the PAM stage, clusters that merge relatively early on, like for example these two here, they merge, and then whatever merges above them cannot be assigned. So you get a big gray blob here of unassigned genes, and sometimes that's okay, but sometimes it's not. You really don't want that. So you turn on PAM, the partitioning around medoids, and these genes then get assigned to the nearest cluster, which may not actually be one of these two. It may be, one of these, and so on. Okay, so a lot of the genes that here would be gray will be assigned to the nearest cluster, which is often advantageous, but sometimes not. I know people who don't use PAM stage, period. Yeah, question?
More of a practical question. I do understand these two settings that more of a cluster is also not good as a cluster. So how do I get a feel for event data that what is the correct way of clustering? If back and forth doing multiple iterations, what do you feel like is the correct way?
So I'll just repeat the question. The question is, I guess that it's not clear what the best way is to cluster, whether you should use PAM or not, whether you should do deep split 1, 2, or 3. And it's a very good question, and I don't have an answer to it, except that when I get the modules, I look at whether they look similar in terms of their functional annotation, whether the module looks similar in terms of how they relate to sample traits. And if I see modules that look very similar in terms of their functional annotation, and they look very similar in terms, they relate similarly to the sample traits that I have, then I think, okay, maybe these two should be merged, and I should sort of decrease deep split a little bit. I've oversplit the modules. On the other hand, sometimes, especially by looking at the dendrogram, I can see, there are two branches that look really different, and I have reasons to believe that, they contain different genes. So I try a higher deep split. And with the PAM stage, it's also, I use it most often because otherwise too many genes would be left out. But again, it's
Also depends on the sample size.
yeah, it really depends on.
You have hundreds of samples, higher needs.
Yeah. So it also depends on number on as Steve says, it also depends on the sample size or on the overall confidence that you have in the data. If you have hundreds of samples, then even relatively fine splits are likely to reflect, true differences in the data. If you only have 20 samples, there's a much higher chance that what you see is just noise in the finer splits, and only the bigger splits really make sense. Yeah. Mm-hmm. Mm-hmm. Yeah. So I guess the question is, if you do the analysis several times and one of the times you get a really good you get a module that has a really high correlation with the phenotype, and other times you don't, and you pick, of course, you pick the one where you have the strong correlation, is that manipulation of the data, is the data dredging, and I would say it is a little bit, but as long as you don't do it hundreds of times, as long as you try 2 or 3 times, and then I mean you can report it in your manuscript. You can say, well, this module is relatively fragile because we only found it with one setting of the arguments, but not with other settings. The other possibility is just to do multiple testing correction. You could think, I did it 3 times, so I multiply all the p-values by number 3. Then it's more than conservative, so that's more than enough to correct for it. We usually hope that the results, and Steve will show you, that the results you get are relatively robust. And so if you change the parameters a little bit, you shouldn't be finding huge differences. That's my comment to it.
And don't walk away thinking that this is very unstable. The opposite is the case. Look at this plot. I mean, the colors show very high agreement. In other words, any way you cut the branches, you get the same output. That's how I look at it. And also remember, the key thing we're after is really the module eigengenes. It's not the modules, it's the module eigengenes. And they turn out to be hugely robust. You can throw out half the genes or 2/3 of the genes inside a module, and the definition of the module eigengene is very effective. One thing you didn't say was, what are the sorts of things that are used to measure this similarity?
Yeah, so the question is, what are the sorts of things that are used to measure the dissimilarity? And that's an excellent question, but unfortunately, I only have 30 minutes for this talk, so I didn't want to go into the detail. But so let me go back a little bit and just answer the question very quickly. There are many things that go into it, but the most important ones, or the most simple ones, are you basically you take what I call the core of the branch, which is a certain number of objects that have been the first merged into the branch. And you look at their average distance, which is roughly this number, the height of this line. And then you look at the height of where the 2 branches merge. And the difference between them I call the gap. It's sort of and the gap tells you how optically or visually how well the branches are defined. And so if this gap is big enough, the branches can be called separate. I also require that the core is far enough from this red line. Okay, if the core is just barely below the red line, that's not a good branch for me. On the other hand, if the merge is above the red line, then it's a good merge. I mean, if if the branches merge very high, it means their average dissimilarity is very big, then that's good enough. So there's a multitude of criteria, and you can fine-tune them, and I just don't have time to explain them all. So
I was really asking, what are the measurements on the
Oh, sorry. So for that, I will go back to here. So remember, you start with the dissimilarity matrix, and those are numbers. And so for each merge, the number, the height of the merge tells you the dissimilarity here. So for two objects, for example, these two objects, the height, which is 0.65, basically gives you the number between the gene 5 and gene 6 here in the matrix, which is 0.5 or 0.6. Oh, okay. So yeah, so that depends on the application. In a correlation network analysis, roughly speaking, you take the correlations between two, let's say the correlation between two genes, okay, and you transform it in many ways, but the simplest way to think about it is that correlation is a similarity measure. So high correlation means very similar. You take 1 minus the correlation, becomes a dissimilarity. Okay, it's too simplistic for our applications, but that's basically how it works. Okay, so we're back to, we were here at PAM stage, and so that's actually the last slide. So the take-home highlights, or the take-home message that you should remember from this talk, is that hierarchical clustering is a useful method for finding groups of similar objects, and it works well in high-throughput data and for exploratory analyses. It produces a hierarchical clustering tree, or a dendrogram, which is useful because it allows you to visualize the cluster structure in the data. Clusters are the branches of the tree, and cluster identification is also known as tree cutting, or branch pruning, or branch cutting, or perhaps even other phrases. There are simple methods for cluster identification which are very intuitive, but they don't always work well. They're not always suitable. So more complicated methods like the dynamic tree cut are capable of identifying clusters in more complicated trees. I think I miswrote here a little bit. In R, you would use the dynamic tree cut library and the function cuttree dynamic. And the most important options or arguments for this function are deep split and PAM stage. So those are the two ones you should remember if you want to remember anything. But the good news is that a single setting for deep split and PAM stage works reasonably well across many applications. It produces comparable results, and which means that the dynamic tree cut is much more suitable for automation than the simple static tree cut method. Okay, I'll stop here. We're a little bit less ahead of the schedule, but
we should discuss a little bit what if you have a data set with 200 variables, like microRNA data. How would you do cluster definition? Because my point is, so the minimum cluster size is another important parameter. So when you have only 100 variables, you might be interested in clusters comprised of 5 or 6 objects.
okay, so Steve makes an important comment, and that's that in the cluster, among the arguments of the cut tree dynamic function of the dynamic tree cut, but also this is also an argument for the static method, is the minimum cluster size that you want to enforce. And in RNA-seq or in gene microarray data, where you would typically have about 20,000 variables, a minimum size of 20 or 30 may be appropriate. Now, if you're working with, let's say, microRNA data, where you only have maybe 200 or 300 microRNAs, the minimum size of 20 or 30 may be too big, so you want to, change it to maybe 5 or 6. You could, I don't know, there could be other, even smaller, perhaps, data sets where the minimum size could be as low as 3, but yeah, that's a good point. Yeah, so in this, and the question was whether we're using a correlation-based, yes? Correlation-based dissimilarity.
Correlation-based dissimilarity metrics are, I think, it's very high intense, so you sort of see these. What if I'm using a regression-based metric or a very sparse regression-based metric, and then the topological matrix itself is very sparse? Still, the hierarchical clustering works better for it, or should I have to look into some other combination?
Okay, so the question is, since we're using a correlation-based metric, the correlation-based similarity tends to be fairly dense, so there's a lot of relatively strong correlations. But there are other methods that produce a much more sparse similarity matrix. So there are relatively few pairs of genes that have a large similarity or a small dissimilarity. And the question is whether hierarchical clustering or perhaps the dynamic tree cut still works in such cases. I think hierarchical clustering can still work. You may want to explore a different version of hierarchical clustering. So the average linkage may not be appropriate, but for example, a single linkage may work better if it's a very sparse matrix. If you have a clustering tree that has well-defined branches, it doesn't really matter that much what kind of method was used to arrive at it. The dynamic tree cut should work relatively well, although I should say that there are certain quantities computed by the function you do use averages. So they're sort of more consistent with the average linkage hierarchical clustering than they would be with the other methods. But as long as the tree structure looks anything like branches, it should still work fairly well. Yeah.
Thank you all. We have now a 15-minute break. And I will talk today about weighted correlation network analysis, which also is known under its